Hello friends, I'm Shiribanu from TutorCabin. Today in this lecture we discuss about the DNA. DNA is a part first of the chapter number 6. It is a molecular process of inheritance. So let's start it. What is the DNA? DNA is a genetic material, okay? Because DNA carry transmitted genetic material and transmitted from one generation to the another generation. We will discuss our last lectures of chapter number 5. We discuss about the gene and chromosome. Both are made up of DNA. Clear? So DNA gene is also a small segment of DNA. Clear? So DNA actually express the character also, and DNA contain a particular character, particular information. It means genetic information, and they are transmit from one generation to the another generation. DNA is a group of molecules that is responsible because DNA is a group of molecules. It contains so many different types of molecules. Clear? Responsible for carrying and transmitting. They contain the information and transmit from one generation to the another generation. The heredity material. So, because they are carrying and transmitted, it is known as a heredity material. That's why they are carry and transmit from one generation to the next generation. Because they are long polymer of ribonucleotide, deoxyribonucleotides. Length of DNA usually defined as a number of nucleotides present in it. Because the length of DNA is mainly proportion to the number of DNA are present or not. So, if more and more number of DNA nucleotides are present, so the DNA will be long, clear or a lengthy. But in case of less amount of nucleotide or less number of nucleotides are present, so the DNA becomes short. Clear? Length is usually defined as a number of nucleotide present. Clear? There are three types of DNA: aDNA, bDNA, and zDNA. In 1869, Johannes Friedrich Michel was first identified and recognized DNA during his research work on WBC, means white blood cell. Mixture was first recognized and identified in 1969 DNA as a nuclein. They are named as nuclein because they recognize as the acidic substance. Some acidic substance are present in the nucleus, that is called as a DNA. They are recognized on his research during his research work on WBC. Second thing, in 1953, Watson and Crick both are proposed the model of DNA double helical structure or double helix model of DNA. With the help of X-ray graph diffraction data. With the help of X-ray diffraction. X-ray diffraction is a technique which is used in the biotechnology. Clear? These methods are helpful to recognize the actual model of the particular component. So, by the help of X-ray diffraction data which is produced by Wilkins and Franklin. Here, both the scientists are worked on X-ray and they have produced some data, and these data are applied for the detection of the model of DNA double helix. By the help of these data, Watson and Crick proposed the model of DNA double helical structure or double helix model. Okay, so many question arises on the basis of who was proposed the DNA double helical structure or model. Watson and Crick proposed the model of DNA double helical structure. Okay. Composed of DNA may be composed of three different components such as sugar, phosphate, and nitrogenous bases. Clear? So the three mainly components are very important. There is a sugar, nitrogenous bases, and phosphate groups. Clear? The main four nitrogenous bases are adenine, thymine, guanine, and cytosine. Clear? All these are very important and basis of DNA. Clear? The arrangement of DNA is this type. These structure are define the arrangement of DNA. So friends, please see this. These circled structure are defined as a phosphate group are attached here, and this is a some capsule type structure define the deoxyribose sugar arrangement. Both are the backbone, they are support as a backbone of base pair. So this is a middle structure, this is a pentagon structure are defined as a nitrogenous bases. These nitrogenous bases are joined with each other with the help of double or triple bond of hydrogen bonds. Clear? So the purine and pyrimidine are form nitrogenous bases. There are two types of purine and two types of pyrimidine. One first, purine are A and G. Adenine and guanine are pyrimidine, and purine and pyrimidine are thymine and cytosine. Both are present in DNA in equal proportion. It is proved by the Chargaff rules. William Chargaff proposed the rule 1950, and they are proved that both purine and pyrimidine means AG and TC, adenine, guanine, and cytosine thymine are present in DNA in equal proportion. So this type of data expresses the equal proportion. The ratio is 1 is to 1. So it's proved that both are present in equal proportion by the rule of Chargaff's. Clear? So many questions are also arises on this rule. So please note it down this thing. So the structure of DNA is important. So the important thing is nucleoside. It means sugar and nitrogenous bases. If sugar plus nitrogenous bases are present, it is called as nucleoside. If nucleoside means sugar, nitrogenous bases plus phosphate, all three things are present. So this is called as a nucleoside. Okay. So friends, let us see. This is a structure of DNA. Clear? 5 prime, 3 prime, 5 prime to 3 prime, 3 prime to 5 prime, structure 5 prime to 3 prime, 3 prime to 5 prime, structure of DNA. These both are the base pairs. Clear? This is the base pairs. And the cytosine are attached with guanine. Here guanine attached with cytosine. Clear? Adenine attached with thymine, and here thymine attached with adenine. So these are the nitrogenous bases, and this is a some line black line structure are shown as a sugar plus phosphate backbone. Is here phosphate plus sugar. So one side is sugar and one side is some arranged like this, and this sugar and phosphate they are backbone, and they are support to the nitrogenous support provided to the nitrogenous bases. So here is the backbone of sugar, and this is a 2 nanometer lengthy, and the length of DNA depends on the presence of number of nucleotides here. So this is a total length of the DNA is 3.4 angstrom, and the sum this the length between the two base pair is 0.34 nanometer. So there are the mainly there are so many. This is a twisted structure. If the twisted are occur deeply, it's called as major groove, and if superficially, so then it is called as a minor groove. So all thing is covered, but one thing is remaining. What is it? It is central dogma. It means DNA are involved and play a very vital role for the production or synthesis of protein. Yeah, it is true. Because DNA are prepared or make our self copy with the help of replication process. So these DNA are form of a copy, our self copy. It is known as a replication. Replication means produce our own copy. Clear? Same to same copy. Clear? That is called as the replication. DNA are triggered the mRNA. There are three types of RNA: mRNA, tRNA, and rRNA. So the RNA are triggered by the DNA by transcription. This process is the transcription. The signals are gone to the RNA from DNA, mRNA. This is the messenger RNA, and these messenger RNA recognize the signal and then transfer to the rRNA for the synthesis of protein. So if the DNA form RNA, the DNA are formed RNA with the help of transcription, and RNA formed protein with the help of translation. So DNA are involved in the production of clear. So friends, this is clear in this chapter. Denucleic acid is helpful to prepare the group of DNA is a group of molecule responsible for carrying and transmitting and the hereditary material contain information and transfer from one generation to the next. This is a long polymer. Length of DNA usually defined as a number of nucleotide present. Yes. So there are three types of A, B, and Z. Mainly B are present in humans. 1869 gems which are proposed firstly identified and recognized the DNA during his research, and they are called as a nuclein, the acidic substance present in the nucleus. In 1953, Watson and Crick proposed the model DNA, the double helical structure with the help of X-ray diffraction. Wilkins and Franklin produce the X-ray diffraction data. Three main component: sugar, phosphate, and nitrogenous liquid. Four bases: adenine, thymine, guanine, and cytosine. Phosphate, nitrogen, and deoxy. This is the structure of mainly this type of structure are shown in the DNA. Two purines and two pyrimidines. Adenine, guanine, purine, and thymine and cytosine are pyrimidine. Chirgaff rules. Erwin Chirgaff proved that the proof by the rule means A, G, and TC adenine, purine, and pyrimidine are present in equal proportion. Are proved the help of Chirgaff rule. Here, so this is the structure of DNA, and this is the central dogma. It means DNA forms the RNA, and RNA forms the protein, and these signals are from transfer from one from to here. Clear, friends? So I hope you understand in this lecture. So please note it down all the important points. So friends, you study well in this lecture. If you have any query, please ask me. Thank you.